Hello everyone. Our next uh, chapter is immunology. First of all, you should know what do you mean by pathogenicity. Pathogenicity means the ability of the microorganisms to cause disease in a host. So it's the ability of the specific microbial species to cause disease in a host. For example, the pathogenicity of species Streptococcus pyogenes, it is the ability to cause throat and other infection, right? So other term is virulence. So virulence on other hand, it is measure of degree or intensity of the pathogenicity of particular strain. So it relates to how severe the disease caused by specific strain of microbe can be. For example, some strain of the influenza virus they cause mild illness, while the other may lead to severe and even life-threatening infection. So this virulence of the strain it is influenced by genetic factors like presence of certain virulence genes. The two terms that you should know, what do you mean by exaltation and what do you mean by attenuation? Exaltation, it is the enhancement of the virulence. So it is a process by which the virulence of microbial strain it increases. It often involves acquisition of new genetic elements or mutation that enhances the microbe organism ability to cause disease. So viral exaltation, it's the measure of increase in the virulence. It is process by which the virulence of microbial strain it increases. Whereas attenuation is the decrease in the virulence. So attenuation it is a process of reducing the virulence of the microbial strain. The method includes passing the microbes through unfavorable host, repeated culture in artificial media or subjecting to high temperature, etc. Other terminology that you should know is what do you mean by toxigenicity. So toxigenicity is the ability of the certain bacteria to produce toxins that can cause harm to the host. So bacterial toxin they are key factor in the pathogenesis of many infectious disease. Now important is what are exotoxin, what are endotoxin, and what is the difference between them. Exotoxin they are the protein produced and secreted by a certain species of bacteria. So exotoxins they are produced by both gram positive and gram negative bacteria, whereas the endotoxin they are produced by the gram negative bacteria. The exotoxin they are protein, whereas the endotoxin they are the lipopolysaccharides that fragment off. The example of the exotoxin include the tetanus toxin and botulism toxin. So exotoxin they can be treated with formaldehyde to yield toxoid, which are non-toxic. Exotoxin they are highly antigenic, whereas the endotoxin they are poorly antigenic. Exotoxin they are heat labile, whereas endotoxin they are heat stable. Endotoxin, they are heat stable lipopolysaccharides that are the part of gram negative bacteria. So toxicity, their toxicity primarily depend upon the lipid component that is known as lipid A. Endotoxin, they are not actively secreted by bacteria but are released when the bacterial cell it disintegrate. Unlike the exotoxin, endotoxin cannot be converted into toxoid. Endotoxin, they can cause various effects in hosts including fever, leukopenia, thrombocytopenia. So IV administration of large dose of endotoxin can lead to endotoxin shock, that is a potential life-threatening condition. Other terminology that you should know is what is bacteremia. Bacteremia refers to the presence of bacteria that are circulating in the bloodstream. It means that bacteria from a local infection or another source have entered the bloodstream, but it does not necessarily imply a widespread systemic infection. What is septicemia? Septicemia, it is more severe than bacteremia. It refers to presence of bacteria in the blood like the bacteremia, but it also involves the multiplication of these bacteria in the bloodstream. So in addition, the septicemia can lead to production of toxic products by the bacteria and it is associated with high fever. So septicemia can be life-threatening as toxin and bacteria circulating in the bloodstream can have a harmful effect on various organs and systems in the body. What is pyemia? Pyemia, it is caused by the pyogenic bacteria. Pyogenic bacteria are organisms that are known to form pus. So these abscess, they are localized collection of pus and hallmark of pyemia. So these are the etiology that you should know. Now let us discuss about the immunity and the types of immunity. The immunity can be innate immunity or adaptive immunity. The adaptive immunity can be natural or artificial. Natural can be subdivided into artificial and passive, whereas the artificial adaptive immunity, it can be divided into again into active and passive immunity. So the innate immunity, remember, it serve as body primary defense against the infection, offering immediate first defense against the infection. So innate immunity, it is also known as natural or native immunity. So this innate immunity can be considered at different levels, including species, races, or individual. So the innate immunity, it's the natural or native immunity. It consists of 4 types of defense barriers, that are the anatomical barriers, phagocytic barriers, blood proteins, or cytokines. Anatomical barriers includes the physical structures and characteristic of body that act as a first line of defense against the invading pathogens. Example are the skin, mucous membrane, and various structures in the different part of the body. like the respiratory, digestive, and urinary tract. So these barriers would act as a physical blockage that prevents many pathogens from entering the body and causing infection. For example, the intact skin, it acts as a barrier to prevent pathogen from entering through the outermost layer. Second is the phagocytic barrier. It involves the action of phagocytic cells like macrophages, neutrophils, and dendritic cells. So these cells, they are specialized immune cells that are capable of phagocytosis. So that means they can engulf and digest the foreign invaders like bacteria and other microorganisms. The third are the blood protein in innate immunity that includes components like uh, complement protein that are part of the complement system and various uh, acute phase proteins. Acute phase proteins, they are produced by liver in response to infection or inflammation, whereas the complement proteins, they are group of serum protein that play a role in enhancing the immune response. The last are the cytokines. Cytokines, they are the signaling molecules that are produced by immune cells. They play a role in regulating the innate immunity. So cytokines have various functions, including activating the immune cells, promoting inflammation, and coordinating the body response to infection. So innate immunity will rely on these four barriers. Defense barriers to provide immediate and non-specific protection against the pathogen. Now, what are the factors that affect the innate immunity? The first is the age. The very young and elderly, they are generally more susceptible to infectious diseases. The fetus in utero, it is protected from the maternal infection by the placental barrier. But sometimes pathogen, they can cross the barrier and lead to fetal infection. The second are the hormones. Hormones can affect the susceptibility. Example, the onset of puberty can lead to spontaneous uh, cure of the infection. Some infections like poliomyelitis, chickenpox, they can be severe in adults than in children due to the hypersensitivity reaction. Endocrine disorders like diabetes, hypothyroidism, adrenal dysfunction, they can enhance the susceptibility to infection. Corticosteroids, they can suppress the host immunity. Elevated sterol level during the pregnancy may increase susceptibility to infections. The third is the nutrition. Malnutrition has complex relation with the immunity. So severe protein deficiency can lead to negative responses in the cell mediated immune test like the Mantox test, etc. Now, what is acquired immunity? Acquired immunity, it is the immunity that is developed by host after exposure to suitable antigen or after transfer of antibodies from immune donor. Adaptive or acquired immunity, it is the resistance that an individual develops during the lifetime by recognizing and selectively eliminating the specific foreign molecules or the antigens. The acquired immunity will depend upon the four factors. The first is the antigenic specificity. So, antigenic specificity refers to the ability of the immune system to distinguish and respond to specific foreign molecules known as antigens. Like in case of infection with the measles virus, the immune system recognizes and specifically targets the antigen on surface of virus. It does not respond in same way to unrelated pathogens like the flu virus. So it is like your immune system ability to recognize and react to specific enemies. It is as it knows who the bad guys are. Think of measles virus as one of these bad guys. So your immune system can spot the specific signals on the measles virus and fight it, but it won't react the same way to the other viruses like the flu. So that is antigenic specificity. The second is diversity. Uh, consider the influenza virus, which can mutate over time. So imagine the flu virus that changes its appearance over time. So your diverse immune system can it can mutate over time. That means it can change its appearance over time. So your diverse immune system, it can create new soldiers, that is the antibodies, to deal with each new look the virus takes on. So it's the immune system diversity that allows it to generate new antibodies to recognize and combat different strains of the virus as they evolve. So that is the diversity. The third is the immunologic memory. So when an individual, suppose, is exposed to chickenpox virus for first time, their immune system generates a primary response to fight the infection. If they are if they are exposed to same virus again in future, the immune system memories allows it to produce a secondary response, which is more efficient at eliminating the virus. So that is the immunological memory. It is the capacity of the immune system to remember a stronger, more specific and faster response on encountering the same antigen for second time. The third is the self or non-self recognition. So it's the unique feature of the immune system enabling it to distinguish between the body's own molecule and foreign antigens. Example, in the autoimmune diseases like the rheumatoid arthritis, the immune system would fail to maintain self-tolerance and it mistakenly targets the body on joints, tissues as it weakens as if they were foreign invaders. So that results in inflammation and damage to the joint. So this is a type of immunity that develops in response to exposure to specific antigens and is characterized by immune system ability to recognize and respond to particular pathogens or the foreign substances. So it helps to distinguish between the body own molecules and the foreign antigen. So these are the four characteristics of the acquired immunity. Now acquired immunity can be of two types. One is active and other is passive. The active can be of two types, natural active or artificial active. So the active immunity, it is type of immunity that occurs when the individual immune system it is exposed to pathogen or antigen that triggers the production of immune response. So these are of two types, natural active. Natural active immunity occurs when the person it is exposed to live pathogen through natural infections or contact with the pathogen in the environment. So over the time, the immune system remembers how to fight that particular pathogen, providing a long-lasting protection. For example, if someone contacts and recover from chickenpox, the immune system develops natural active immunity making the making them immune to further chickenpox infection. The second is the artificial active immunity. The artificial active immunity occurs when the individual it is intentionally exposed to weakened or inactive form of pathogen, usually through vaccination. So artificial active immunity it is type of immunity that is induced by the vaccine. Vaccine they are prepared containing either live or killed microorganisms or their product that are used to stimulate the immune system. So the live bacterial vaccine, the example is BCG. So it is used for tuberculosis. The example of killed bacterial vaccine, it's the cholera. The example of bacterial product vaccine, it's the tetanus toxoid vaccine that contains inactive bacterial toxins. The example of live viral vaccine, it's the oral polio virus, that is the Sabin vaccine. So that contains live but weakened polio virus. The example of killed viral vaccine, it's the Salk vaccine. It's the injectable. It's the injectable polio vaccine that contains inactive polio virus. So the live vaccine typically provides long-lasting immunity that can last for several years. However, booster dose they may be necessary to maintain protection. Killed vaccine, on other hand, they are generally less immunogenic, mean they produce a weaker immune response compared to the live vaccine. Protection for the from the killed vaccine usually lasts for a shorter period, so booster dose they are often required. The initial dose is the primary dose and subsequent doses they are the booster doses. Now the passive acquired immunity, it is type of immunity that occurs when individual it receive preformed antibodies from an external source rather than producing these antibodies themselves through their own immune response. It can be natural passive immunity or the artificial passive immunity. So natural passive immunity or the artificial passive immunity. So the natural passive immunity it occurs when a newborn baby he acquires antibodies from the mother. So these antibodies, these maternal antibodies, they are transferred to baby during pregnancy through the placenta and through the breast milk during breastfeeding. So the transferred antibodies they provide temporary protection to the infant against specific infection until their own immune system matures. So, for example, a mother who has immunity to particular disease like the measles can pass on the protective immunity to her baby. So, these maternal antibodies protect the baby from measles during the first month of their life. Other is the artificial passive immunity. So that involves the administration of preformed antibodies or immune components that are derived from the external source. The example is the use of the immune globulin. So it is a preparation of antibodies to provide temporary protection against the diseases like the hepatitis A, rabies, or the tetanus after the exposure to the pathogens. So the source of the antibodies in the artificial passive immunity include the hyperimmune sera or convalescent sera. So hyperimmune sera, these are the sera that is obtained from the animals or humans who have been exposed to vaccine against a specific pathogen to generate a high level of antibodies, like the anti-serum, like the anti-tetanus serum that is obtained from the hyperimmune horses. Whereas the convalescent sera, these are the sera that is collected from individuals who have recently recovered from specific infectious diseases. So they contains a high level of antibodies against the pathogen that are responsible for disease, like the pooled human gamma globin. So it is a preparation of gamma globin, a component of blood plasma collected from pooled sera of the healthy adult donors. Now, this is the difference between the active and the passive immunity. The active immunity it is long lasting, whereas passive immunity it is short term. Uh, so, active immunity it is induced by infections or immunogens, whereas passive immunity it is confirmed by administration of ready-made antibodies. Active immunity has immunological memory present, but in passive immunity no memory is there. Now, other important term is a local immunity. So it was performed by Bafredka. That is concept that is importance of the immune response at the site of the entry of pathogen in the body. So local immunity it is site specific immune response. Like the gut mucosa and respiratory mucosa, they are the common site of the pathogen entry. Example of the poliomyelitis. In case of poliomyelitis, the active immunization with the killed vaccine provides a systemic immunity. So that means it can neutralize the virus once it enters the bloodstream. This type of immunity may not prevent the initial multiplication of virus at site of entry like the gut mucosa. So the local intestinal immunity that can be acquired through the natural immunity with the live oral vaccine, it is important for preventing the viral replication at point of entry. Other is the role of IgA antibodies. So local immunity often involves the production of specific class of immunoglobulins that are IgA. Secretory IgA is one of the IgA antibodies that is produced locally by the plasma cells present on the mucosal surface or in the secretory glands. So the local immunity it is important in protecting against a pathogen that primarily enter the body through the mucosal route, like the respiratory digestive tract. Whereas the herd immunity, it is also known as community immunity or the population immunity refers to the overall level of immunity within community or the population. So it occurs when a large population of individuals in community become immune to particular pathogens like virus or bacteria.